Here we'll review the insulin structure, synthesis, and regulation in preparation for discussion about glucose regulation and diabetes. So you might remember that insulin is a member of the peptide or protein class of hormones. And a peptide hormone is a water-soluble hormone made up of a few amino acids that introduces a series of chemical reactions to change the cell's metabolism. So insulin starts out as a precursor molecule, which is converted over a series of steps from pre-proinsulin to proinsulin, and finally to the active insulin molecule. Let's outline the steps involved in the generation of insulin from its precursor proteins. So note that all of these steps are happening within the beta cell. And in step 1, a signal peptide sequence on the pre-proinsulin molecule directs it to the endoplasmic reticulum. Then in step 2, pre-proinsulin is converted to proinsulin simply by loss of that signal sequence, which is shown in blue. In step 3, the newly formed proinsulin exits the endoplasmic reticulum, transits through the Golgi, and enters the secretory vesicles. Step 4 occurs in the secretory vesicles. C-peptide, the connecting peptide, which is shown here in green, is cleaved. And so you're left with a alpha chain here in yellow and a beta chain here in red, which together form the mature insulin molecule. And this molecule is stored within those granules and is ready for release upon the proper stimulus. This is a really basic schematic of insulin structure. And so insulin's a relatively small protein weighing about 6 kilodaltons, and it's made up of those two chains, an alpha chain and a beta chain that are held together by two disulfide bonds. Now that we've learned about insulin structure, let's see how it's regulated and released, starting with what stimulates insulin release from the beta cell. And the major stimulus for insulin release, as you might expect, is a rise in blood glucose. And this occurs in the fed state or after nutrients from a meal are absorbed from the gut into the bloodstream. However, there are also lesser non-glucose stimuli for insulin release. And these stimuli include amino acids and acetylcholine, and they're not as potent as glucose, but they're still important in specific states. The incretins are also listed here. They're peptides that are released from the gut in response to meals before those meals are even digested or absorbed into the bloodstream. So you can think of these as kind of warning signals that the blood glucose is about to rise, and therefore this incretin-stimulated insulin release is a great way for the body to ensure that insulin is available in the bloodstream even prior to the post-meal rise in blood sugar. And the importance of incretins will be covered in much greater detail during the discussion on pharmacologic options for treating type 2 diabetes. But for now, just know that there are analogs of incretins that are very effective therapy for controlling type 2 diabetes. So since glucose is the primary stimulus for insulin release, let's review the mechanism by which this process occurs. So here are the steps just listed sequentially, and coming up we'll see these steps on a cartoon image of a beta cell. And so the main steps are step 1, where glucose enters the beta cell, and step 7, where insulin is ultimately released after this series of steps within the beta cell. So now let's look at the visual depiction of this process. So here we're looking at the beta cell with its plasma membrane and multiple transporters embedded within. So in step 1, glucose enters the beta cell via GLUT2 transporters, and these are a facilitated glucose transporter. They also exist in the liver, and the kidney, and the brain, in addition to these pancreatic beta cells pictured here. These GLUT2 transporters are important because they ensure a large bidirectional flux of glucose into and out of the cell due to their low affinity and high capacity. So the beta cell, in essence, can easily sense from minute to minute the glucose level in the bloodstream. And later on, you'll hear about other types of glucose transporters that allow for glucose uptake in other cells. So once glucose enters the beta cell, ATP is generated by glycolysis. Then ATP-sensitive potassium channels in the plasma membrane will close, allowing that plasma membrane to depolarize, which eventually allows the voltage-gated calcium channels to open. Calcium entering the beta cell is the stimulus for those insulin granules inside the cell to fuse with the cell membrane and then release insulin into the bloodstream. And as an aside, you'll note that there's a separate receptor called a sulfonylurea receptor, shown here in yellow next to the ATP-sensitive potassium channel. And the reason it's called the sulfonylurea receptor is because it allows for a class of drugs called the sulfonylureas to bypass the glucose sensing mechanism of the beta cell and directly allow insulin release into the bloodstream. Again, a useful class of drugs when trying to treat type 2 diabetes. So now that we've seen how insulin is released from the beta cell, let's think about the action of insulin on its target cells. So after insulin is released, it travels through the blood and binds the insulin receptor on target cells, which is a tyrosine kinase. And upon binding its receptor, insulin activates the RAS and PI3 kinase pathways in the target cells, and these are mainly in liver and muscle and adipose cells. And the bottom line is that insulin acts to reduce blood glucose levels, and it does this by promoting different metabolic changes in each type of target cell. Now let's look at the insulin receptor and the effect of insulin binding in each target cell type individually. So the insulin receptor is depicted here, and there are 3 key features to remember about this receptor. So first, it's composed of 2 alpha and 2 beta subunits. The alpha chains are yellow and are extracellular, so they're outside the cell, and they contain the insulin binding domain. So insulin will bind here. The beta chains penetrate the plasma membrane, and they are the ATP binding sites. The whole receptor itself is a tyrosine kinase, as we mentioned, and this means that it's an enzyme that transfers phosphate groups from ATP to intracellular proteins. So once insulin binds to this alpha subunit, it will cause the beta subunit inside the cell to phosphorylate themselves or autophosphorylate, and this activates the catalytic activity of the receptor and allows for phosphorylation of a number of downstream intracellular proteins to elicit a biological response. So now let's look more closely at the effects of insulin binding on those 3 main target cell types, the liver, the muscle, and the adipose tissue. So starting with liver, remember that the liver has the ability to both produce glucose and store glucose. So when insulin binds to the insulin receptor on liver cells, glucose production is decreased and glucose storage is increased. Other fuel forms, including fatty acids, are synthesized and stored for later use as well as a result of insulin binding to the liver cells. And this is exactly what you want to happen in this fed state after a meal. Now if we look at muscle cells, insulin binding to the insulin receptors here also causes a decrease in glucose production and an increase in glucose storage, just as we saw in the liver. But notice that something else happens here, and that's that Glucose transporters, which are the same GLUT4 transporters we talked about previously, are translocated from the cytoplasm of the muscle cell to its cell surface, which allows for an increase in glucose uptake into the muscle cell. So this GLUT4 translocation is the primary action of insulin in muscle, and it's really important way to dispose of glucose in the fed state or whenever blood glucose levels rise. And so as a side note, this is one of the many processes that can be impaired in patients who have type 2 diabetes, and it can account for part of that, quote, insulin resistance that's seen in type 2 diabetes. And finally, these are fat or adipose cells, and insulin binding the insulin receptor also increases glucose uptake via the GLUT4 receptors, just as we saw in the muscle cell. In addition, it also causes increased glucose storage, just as we saw in both the liver and the muscle. But a unique and important effect of insulin on the fat cell is to decrease the release of free fatty acids from those fat cells into the bloodstream. And at the same time, insulin will increase the storage of those free fatty acids as triglycerides within the adipose tissue. So this triglyceride storage is promoted by increasing LPL or lipoprotein lipase activity. So it should be evident by now that even though insulin is best known for its role in regulating blood sugar, that's certainly not insulin's only role. So we just saw that insulin promotes protein and fatty acid synthesis in the liver and muscle, and it also promotes the storage of triglycerides in adipose tissue. And the end effects of all of these processes is that there's less substrate of any kind that's available for the liver or for other organs to shunt into glucose production or gluconeogenesis. So in the end, insulin not only allows for post-meal glucose to be taken up by the muscle, liver, and fat cells, but insulin also inhibits these cells from providing substrate to make new glucose. And this concept will become important later during discussions about hyperglycemic emergencies in which one or more of these processes is disturbed. And this image just shows the big picture. On the left is the beta cell where insulin is produced and released right beside one of the three main target cell types. In this case, it's a muscle cell. And this system is a really elegant one where the pancreatic beta cell can easily sense a rise in blood glucose and fairly quickly respond by releasing the preformed and pre-stored insulin. The target cells, in turn, bind insulin via their membrane-bound insulin receptors and then trigger a series of phosphorylation events within the cell. And ultimately, there's increased uptake of glucose from the bloodstream, increased glucose storage and utilization in the cell, and decreased production and release of glucose from the cell.